I'm Peter Kolchinsky, Managing Partner of RA Capital Management. I'm an investor, a scientist, and a food allergy parent. So food allergies are personal for me, as they are for many. And in each episode of this monthly podcast, I'll be speaking with a dynamic thought leader about the grand human quest to conquer food allergies. We'll explore the science of food allergies and technologies to prevent, diagnose, and treat them, as well as the complexities of public policy, insurance, business models, and simply human behavior that, like many entrepreneurs know, can kill seemingly good ideas. So join me and let's talk to the brightest minds on the planet about the food allergy solutions we already have and what's on the horizon. Welcome to Future in Sight. All right, we've got another fun episode of Future in Sight. And for this one, I'm joined by Nurry Hong. He's a food allergy expert, CEO of a company called Pareto, which is working on microbiome therapeutics for food allergy. That's a really cool space we're going to talk a fair bit about. And he's the former chief of strategy and innovation for FAIR itself, which is hosting this podcast. So Nurry, welcome, and tell us about your journey. How did you come to this space?
Thanks, Peter. Appreciate you having me on and having the chance to have a discussion with you about food allergy and all the other things that we'll get into. And yeah, talking about my journey here, and I appreciate that you call me a food allergy expert. It's weird to think of myself that way, but I can see that with what I've been doing over the past 4 or 5 years here, I've spent time in this space really trying to help understand it and help participate it as an advocate and as a food allergy father. And I think that that's something that you and I share in terms of common goals and reasons why we're here. But professionally, my background, I come from the biotech and life sciences industry. I'm not an MD or a PhD, so I don't wear one of those fancy scientific titles. Notoriously, I'm known as the business guy within the biotech field. So I was a partner with a consulting firm in the strategy consulting space for a few years, from 2010 to 2017. Had a wonderful time really getting a wide, diverse experience working in the industry, working with great scientists and leaders within that field, touching all topics related to science and R&D and commercial, development, market access issues and pricing issues, policy, global health, digital health, you name it. But then really wanted to kind of take my energy and focus that on something that I could have more direct impact on. The typical, critique of a consultant is that you advise, but you never do. So I wanted to actually get involved and do. And so in around 2017, I decided to try to take what I could in terms of my background and professional expertise and apply to something that I saw was an opportunity in terms of therapeutic development, which was, the nascent field of food allergy therapeutics at the time.
What drew you to it?
My son has peanut allergy and shrimp allergy and other non-IgE mediated, allergic reactions, not quite EOE, but something else that we'll just call, gastric comorbidities. And the lack of development in food allergy compared to what was going on in immunology more broadly within science was just kind of, head smacking in terms of the amount of frustration that I had. So yes, immunotherapy was on its way, but I knew that we could do much better. And in the pipeline, things like Xolair and other antibodies clearly could have potential, which I'm so thrilled to see is now proliferating. But I saw that space and felt that, in the immuno-oncology space, I may have a smaller impact, but within the food allergy space at that time, I felt like I could do something. So I really just spent my time at risk, and thank you to my wife for allowing me to try that, going after early stage opportunities coming out of academia, trying to figure out ways that I could innovate and just do what I could in terms of developing startup biotech companies. One thing led to another. I got really connected into both the scientific community, but also what I'll call the very active donor and parent community. And that led to a discussion at the time where FAIR was trying to also take the next step in its journey
But
and
actually take a step back. I realize, one of the cool things about really getting to know somebody is trying to relate to those little steps they take when they say, I want to do this. And you said you got connected into, this community, but what does that actually mean? Like, who did you talk to when you were like, I think I'm interested in this? And
that's a funny question, because this is something that is very much practice what you preach. So as a dad, I say this to my kids all the time. If you think you can do it, you can do it. And they hate it, right? They absolutely hate it. This is very much a case of that. I quit LEK, or my strategy consulting job, and decided that I was going to go immerse myself in this field. The first thing I did was call all the key opinion leaders that I could find that would be willing to speak with me. And that was literally an email, or most of the time, just a cold email to all of the leading experts to start asking them questions and going to conferences.
To be clear, a lot of them are academic, right? So technically their emails are in their publications, right? Because if you're in the business world, it's harder. You'd have to go through like LinkedIn, and you don't know whether they would treat you seriously.
That's correct.
So God bless all those email addresses in the papers.
And these people were generous with their time. And, we're talking about some of the well-known names within this field. Very early on in the process, someone said, oh, you really need to meet Dave Bunning. And I said, sure. And I didn't know who Dave Bunning was. And I certainly know who Dave Bunning is now. And for the record, chairman of FAIR and longtime advocate within this industry, or this field. And that was really kind of the beginning of my kind of also more broad introduction into the donor field and the advocacy space, as well as, a professional link, as, based on what Dave does within the, as an investor. So that brought me to a different side of the community, which, was more linked to the professional background that I also had. So these things are both personal in terms of me trying to understand scientifically and therapeutically what I could do for my own child, but then also professionally, how do I go make an impact scientifically, what makes sense, and then mapping that to what I knew from kind of more of a market analyst perspective, what makes sense to start really building on, as we think about a future with, multiple treatment opportunities and better diagnostics.
Yeah. So, Dave and FAIR really go back, to a point where it's almost like this, you go back in time and everything comes down to this one point where Aimmune gets formed. This bold idea of like, why don't we actually just develop, peanut powder as a drug? Before, I mean, it already was being used as a drug, right? But the idea that no, it needs to be actually upgraded to a true FDA approved drug and manufactured, to a high quality or whatever, like there's that insight. And it needs funding. It's going to take a ton of money, right? And it requires people with like a business background, investing background, and at the same time, you need an organization that's going to rally people, to the idea that like food allergy is a thing, and requires organized thought around policy and regulations. And so Dave was there at the origin point. And so you networked your way basically to, some of the people that are founders of this more modern, coordinated strategy against food allergies.
You're absolutely right. I found like I felt like I found a center of it all in terms of the origin point of where we are today. And actually the core that I saw externally as an outsider that said, yeah, that is the initial nugget that will make this real for everyone else. Because then with this very first generation treatment, just to be quite frank, it educates the broader investor community and the broader patient community and physician community of what this condition is, and starts a snowball process potentially, if we take advantage of it, to build on it and do better. And so finding Dave and understanding that that was part of the original plan, that they needed to start somewhere, was just reassuring to hear that there were so many other smart, like-minded people that had already, established this.
So and I'll just get the chief strategy and innovation, person at FAIR.
Well, before I get there, I just want to say that, look, I want to absolutely just put this out there. Having you get involved in this makes me incredibly happy to extend, what we were just talking about. So not to, go down that rabbit hole of a discussion, but I just wanted to actually say that I was thrilled to see when you got involved with FAIR and the study with Ruchi, because those are well, thought out ideas and absolutely deserve the kind of attention that you could put to it. All right, so to answer your question,
I'm going to pull on that thread then and say any great organization is always in a state of being founded, right? And so the way you found yourself to FAIR, and we're going to hear your story on how you integrated yourself into the organization and then contributed to, creating a new company that would pursue a new idea. And later I found my way, to FAIR, indeed we've talked in past episodes, but we'll talk, much more about some of the stuff that I'm much more hands-on involved with in terms of Ruchi's study. And I'm pretty sure we're going to have Ruchi on this podcast. She's awesome. And she's got a new book out. Others that are out there, who share this sort of righteous urge to we've got to solve this problem. We can't just accept it lying down and have that warrior spirit. They should network their way. There must be contact information on FAIR's website. If you ping me on LinkedIn, I'll respond. But if there are people out there that say, yeah, I hate food allergies too, and I want to contribute to the cause, then, Nurry, your story is an example of, how you did it. And I've got my own story of how I also found my way to Dave. The path is short. It's available for anybody.
I agree with that. And, I think that you and I could put some heat on the rest of the community to kind of take this to the next level, because I have some ideas that I'm sure you share in terms of what can be done if we just do it. And there's always barriers as to why things can't happen. But in this case, I don't see why they shouldn't happen. So you asked the question, how to get to FAIR. So I did not have an intent to do anything in the nonprofit space. I really appreciated what FAIR was doing. And from afar, if there was something that I could do to help, sure. But, my focus was on trying to figure out how to apply what I knew, which was, start up biotech and go from there. But when FAIR was going through its transition to build on, AImmune and build on the, the momentum that was coming and trying to take this to the next level of, just advocacy, education, policy shaping, and scientific research, I got a call because I knew Dave. Dave was getting involved again with the organization in a hands-on way. The first time I met him, of course, what I did was the most consultant-y thing possible. I shared a deck of what I thought the landscape for food allergy looked like with, all of the segments of intervention and where, a phased plan of how that might evolve. And he kind of just nodded his head and said, yep. So I think with that in mind, he said, how do you enact that and make that real in terms of, what FAIR could become? Previously, I had also had experience working with organizations like JDRF and, other large nonprofit organizations, and had a lot of connections into that type 1 world, type 1 diabetes world, which really, I think, aside from maybe, cystic fibrosis, has got to be considered one of the, the bellwethers in terms of medical.
Yeah, there's Jocelyn.
Yeah. I mean, and everything that they've done is just tremendous, and is still to this day has ramifications of what could be potentially possible with things like prevention in a chronic disease space. So building on what type 1 has done, I wanted to apply some of those lessons also to FAIR. And it's not just, JDRF, but other leading organizations in terms of what is the next step of public-private partnership as you think about what a disease foundation should be doing to advocate for patients.
Yeah, Michael J. Fox in Parkinson's, the CF Foundation, cystic fibrosis. There's some organizations that when they get going, they're very much about the advocacy, the policy, rallying people, to contribute, raise money, but also they've got some deep nerd level expertise in the research, and they are, hunting around, for the brilliant minds out there that need some support, a grant, here or there, to be elevated and linked together in order to, really kickstart, the serious R&D that's required to make progress. That's what FAIR, has certainly been evolving towards.
And they had the clinical network was something that, that a lot of those organizations would love to have. And it was an asset that, I really hope that they continue to, and they are, building on, which I thought was just a tremendous step up in terms of having an existing capability that they could actually do something with, that's tangible. And additionally, the registry as a starting point, all of these things can continue to get better and expand, but the core assets that they had available to them made sense, that they could build on. And I hope that we can keep it going.
What are some of the assets, when you say the core assets that they had available to them?
I think that a couple of things when I was introduced to the organization and where they wanted to go broadly. One, having the network and really the connection to all of the top minds within the community was important. And they had it within any disease area of research. Of course, you've got a lot of key opinion leaders, scientific experts with strong opinions. And one of the things that was possible within this space was bringing those people together and making sure that you didn't have too many silos and factions going off alone. And when you actually went out and spoke with everybody and shared the common vision, any of those considerations would be put aside, that for the common good and the common vision that what wanted to be achieved, there was something there that you could build on, which was really a testament to that professional community,
Yeah.
and Fair's ability to keep it going. And that was all
people like Ruchi, right? So just to be like, this is a very personal space. Like everybody knows each other. Like we had Wayne Schreffler on, I think, for the first episode, and he's my daughter's physician. We had Kari Nadeau on. In fact, she's got 2 episodes. And this is a very small community. So once you get in the middle of it, and if you can get a Zoom call going with some of the greatest minds, you get to the cutting edge of what's known and what's not known in the field. You get to the controversies where not everybody agrees based on the data. And what's clear is we got to do another experiment to find out what's true. So yeah, having an organization that's got all those people, around the table, it's a really great foundation.
Yeah, and their willingness to participate in this more collaborative clinical network with layers to it, and some a lot of encouraged sharing that I hope to come to fruition, and a willingness to give it a try, and thinking about how to advance research and do it in kind of a best-in-class way. I think that's again a testament to that community for sticking through it and going through changes and ongoing iterations of that network design to get to where it is today, that hopefully we can continue to make it better. That was one major asset. The other asset was just its connection to the patient community. I think it was absolutely the #1 source for patients. There are a lot of other sources of information, but FAIR was still the leader, I think, in terms of just basic patient education. And along with that came some responsibility to represent the patient voice nationally in policy and in other advocacy channels. And I think that that was an asset that someone like Lisa Gable coming in and really bringing what she has and her background and her expertise in those communities was a great fit in terms of being able to do something that's actionable on behalf of patients now, and thinking about that voice beyond just the science that's driving the treatments and the solutions, but there are other types of solutions that can also be devised.
Yeah, I'm realizing that we can probably do a whole episode on the galvanizing of a movement around a given disease, by examining how did FAIR start and evolve, and as it matured, which people joined the organization, how they change it. Certainly Lisa joining really reshaped the organization, gave it a lot of momentum and direction. But how did you come to become the chief of strategy and innovation?
So Lisa was the CEO, Dave was the chairman. I was running around trying to start biotech companies, and Dave said, look, I need somebody to come in and help on the biotech strategy side, to think about how do you do what we want to accomplish on the broader advocacy and organizational side that Lisa's going to really be taking to the next level, and doing that on the medical research. front, and how do we make that real and actionable? Because he also knew the risk that goes into investing into therapeutics and novel R&D. So what can you do as an organization?
So tell me this. I sort of get what innovation means, right? It's like probably staying on top of like cutting edge technology. What does strategy mean? What is strategy as it pertains to FAIR and the food allergy space and you and your consulting background? Make it feel real.
So that's a great question. I mean, strategy can be thought of a lot of different ways. I mean, the most simple way to make it tangible, it's a roadmap. It's something that you can actually follow that will lead you to an end destination of, or an outcome that you desire. So in this case, thinking about the broader vision and mission for the organization. And that's not just me, that's Lisa and the broader organization coming to terms with what they want this to be, for the long term. And then mapping back to what does that mean for the patient in terms of treatments and outcomes and an experience that you can actually make real. So as you think about that, there are many, to use a very consultancy way of thinking about it, many pillars or vectors of ways of doing that, right? So there are consumer-based solutions, and there are educational-based solutions and engagement solutions, and all sorts of things that FAIR can and will get involved in. On the medical side, there are everything from standard of care to actual investment into novel R&D. And so when you get into novel R&D, you're really thinking about tangible diagnostics and treatments or cures that will make an impact.
All right, so I like analogies. I'm always looking for analogies. I'm gonna try this one to demystify what strategy means. If one imagines some point in the future where we have put food allergy into checkmate, right? And this isn't your standard game of chess. It's not just like one king, on the board and all the pieces stripped away, and you just need like a bishop and a pawn or whatever. Like, it is a more complicated problem. And you're imagining we've prevented as much food allergy as we can, but there are still some people who develop it. You got to be able to diagnose it. You got to be able to equip them, with the best possible way to prevent or stop anaphylaxis. You need to make sure that, the epinephrine is available everywhere. That's going to require policy changes because, laws don't yet, mandate, for example, at schools and restaurants. When you imagine that future of what would it look like to have used all conceivable possible chess pieces to constrain food allergy, and then, as you say, map your way back to today and start thinking like, all right, we got to incubate, an effort to advocate for the necessary policy changes we'll need in the future. Like the prevention study that we're doing to show that introducing your infants to all these various kinds of foods can prevent food allergy. Well, how do you make that affordable for people, right? Like you got to make sure that the various programs that help people afford fancier foods, are going to cover these things because they're actually medically useful, right? And that may require FDA buy-in. So, being able to map your way back to where we're today and where do we need to go on all these different fronts, FAIR has very much been doing it. And it sounds like you're the person who helped kick off that vision.
It wasn't me. I really want to, say that I was just a part of a broader team that came up with, the ways that FAIR could make an impact. And there was a lot of work done prior to my arrival. And I really got sucked into it because what I wanted to achieve when I left and try to go on this path was to go make a difference. I always thought about that in a private sector perspective, but had enough experience in the nonprofit sector to see where FAIR could obviously make a difference. And there was something that was common between Dave and Lisa and myself, is that we all looked at the state of food allergy, and we always said that it's behind asthma or atopic dermatitis or other disease areas by easily 10 years, if not decades of sophisticated R&D. Well, what do you do with that? Maybe there's an opportunity to avoid some of the mistakes, learn from those those other areas, and in some ways leapfrog how we're going to go approach it in the area of food allergy. And there's a little bit of that kind of vision or overlay, I think, to everything that we thought about trying to accomplish. And so I think there's why I would love to continue to put pressure on the organization to do as much public-private, funding of research, and to really take that to the next level. And to also, whenever we thought about what you're funding, I think implicitly and explicitly in at least my own decision making was, do you get more bang for your buck for this investment? So can this fund something that's obvious that you can speak to and is easy to communicate? But then if you fund that, does that either place the foundation for additional other research or become additional ways of learning that would enable something else in the future? So really thinking about how you place foundational building blocks to
What's
keep
an
building.
example? Let's make it real, because that was sort of abstract. What's an example of something that you've seen funded that then opened up new options?
Well, I am incredibly thrilled that you're funding the SEED study. I think that's a perfect example of there is foundational research that we know about early introduction, and there is a lot that we do not know. So I think it's a very obvious case to be made that we should be doing studies like SEED and more to really take the next level of early introduction and our understanding of that to proliferate and make that real. Let's also then, what else could you do with that? So we know that there are going to be non-responders. We know that there are going to be people who continue to develop allergy. So of those that are quote unquote prevented or not, how do you do the comparison analysis of the fundamental immune tolerance mechanisms within those populations when you compare them to other studies? FAIR is also trying to bank in a standardized way bio samples from both this study and other studies to be able to do more standardized research. I think that's one example of how
That's right.
you take it's an obvious
Yeah.
situation where you should do this study, but look at what you could also build on top of it, in terms of the other learning that is potentially there.
So
and I think that
let's get into microbiome, because it's right there. You're like 1 degree away from what you're saying, right?
yeah,
We're banking samples. The SEED study is going to enroll like 1,000 some, babies, and they're not going to have food allergy at the outset. They're going to collect samples to be able to eventually later analyze their microbiome, I believe, if I'm not mistaken. If they're not, maybe we should look into how do we make sure we get those samples. And it's not core to SEED's main objective, which is to see whether babies that are fed a whole range of foods like peanut butter and tree nut butters and soy and wheat and all those kinds of things, like intensively, where the parents are coached on the importance of early introduction of those foods and sticking with it, compared to parents that basically are simply told to feed their kid like they are today. You're not telling them not to give them peanut butter. You're just leaving it up to their primary care physician, the pediatrician, to say whatever they say, right? And we're going to find out whether there's a reduction in food allergies after a year or 2 years of this, if those babies are really if you make a strong effort to introduce them early to these foods and consistently. And you're right, towards after a year or 2 years, you're going to see that some kids still develop food allergy, right? And you're going to want to collect their samples compared to the samples all the kids who didn't develop food allergies. And now you're going to look at their what are we going to analyze? I'm going to hand it off to you, to teach us about microbiome and the role that this plays, because it is an offshoot study of the SEED study.
Yeah, and in full disclosure, I would love to collaborate with FAIR and for these samples and to kind of share in mechanistic research related to this topic, but also other studies as well. We can take that offline because I have a lot of ideas how to strike a public-private partnership to do.
I don't really believe in conflict of interest when we're all fighting to solve food allergies. So it's like a common interest in this. Yeah,
no, and I think this is a perfect example. This is a perfect example, though. There is a need to bank samples. There's a need to conduct mechanistic research against those samples, and there needs to be some common framework of how we're going to go about doing that with enough open, ended, sharing of research or allowing enough open-ended research for individual groups to kind of pursue what they want to pursue. So those are the details and how things can get overly complicated, but there is a common I think, path here that different stakeholders can bring different things to the equation for us to really, solve some very interesting questions.
Yeah.
Going to the fundamental question of the microbiome. So just to take a step back, I think most people know what we mean by the microbiome, but what I'm describing when I say that is really the gut microbiome. So the many trillions of cells of bacteria that you have that reside in your gut, and really are a almost a symbiotic other living organism within you that are help mediate everything as core as your gut function, which eventually leads to pretty much every other major system within your body, including your immune system, and metabolism and and other aspects. But
Yeah.
fundamentally, when we talk about the gut microbiome, and the reason people take probiotics is there's this notion that making your gut microbiome healthier, however you want to define that, will lead to better health outcomes because of the linkage to fundamental gut health and immune health. The long-term understanding of this has been a proliferation of studies comparing healthy populations to diseased populations, typically at snapshot points in time, and doing a comparison of what's there, what's not there. And we now know through all of that correlation analysis, something is different in healthy versus disease. I mean, I think that very simply put, we know that the gut microbiome of a healthy individual tends to look different from somebody with a given disease state. And let's take IBD or food allergy for this, in this case, as the example. And what that different microbiome looks like, though, is a very difficult question to answer. So what specifically is missing? What combination things are missing? How does that relate to other things that are going on in the body that lead to this disease state? That's where a lot of the complexity obviously resides.
May I ask just a technical question? When we talk about measuring the microbiome, that's in our guts. What samples do we bank? What are we actually measuring?
Poop.
Poop.
I mean,
poop I believe that's a technical term, actually.
It is.
So,
we are poop experts in the microbiome field. So I can't tell you how many days we talk about, fecal samples, to be more technically correct. Right.
But is that the only and right measure of your microbiome throughout your gut?
Very interesting question. So we'll come back to that. So the big unknown is what actually happens in your gut, because what we get to see is what comes out of your gut. What we don't get to see is what actually happens in your gut, both immunologically and in your microbiome. And you can imagine the complexity that opens up, and we'll put that to a side. But it is not the best way, because there would be better ways hypothetically, but it is the best way that we have to easily do it, and has become the standard. So using fecal samples as the primary sample type to measure gut microbiota composition, it is the way that it's the primary way that we're going to be reliant, or doing this. Now, the questions of how do you do that better become very interesting. And of course, as you think about how microbiome companies and academic researchers are approaching it, everybody's trying to do that better. So how do you, with the limitations that we have, the unpacking that complexity and mapping that more accurately, especially if you think about a live gut environment, is where we are all trying to move. And that gets interesting because that brings in, the best of what you would imagine in terms of immune science and computational biology and, all the rest of it.
Has anybody come up with like a pill you swallow that opens up a compartment like at different points in your gut and samples what's in there? Like that's a Bob Langer kind of thing.
I was going to say, I don't know, talk to Bob Langer because he probably has something. I've seen all sorts of things, gut delivery, devices. We've actually thought about the application of that for different purposes, certainly for delivery of immunotherapy. There's a lot of discussion around different GI devices, but not just, for food allergy. That's being discussed in other areas as well. But GI delivery in and of its own right is an interesting area that's applicable.
Actually, that's intriguing. So if you were to like load up a whole bunch of, peanut powder or whatever into a capsule that bypassed, your mouth and only released in the gut, is that a shortcut to,
I think things,
tolerization?
without getting into details, that things like that have been discussed. But the interesting thing with antigen presentation is kind of that next generation of, very targeted antigen presentation through different adjuvant vehicles. A lot of nanoparticle design and encapsulation and presentation to very specific immune, cell types, or other methods of, inducing tolerance.
Yeah.
And I think that kind of what people are doing outside of allergy, all of those platform technologies could be applied to food allergies so easily, which is another example of,
Yeah.
technology kind of developing in parallel that we can kind of maybe steal from and accelerate what we're trying to do. It's already happening in food allergy. I think it could be taken to another level though.
All right, so we could totally go down all these rabbit holes.
Yeah.
Specifically,
Okay.
Pareto's approach.
Okay, so what we're doing is, okay, so what if somebody wanted to be critical of the microbiome space? Absolutely, you could, because it's very crude still, right? What we don't have a very good understanding of is that PK/PD relationship, or for the layperson, how to dose for the maximal effect, in terms of either a wide consortium of bacteria, a very specific, defined set of bacteria, or something that comes from bacteria in terms of other, byproducts. Now, once you get to metabolites and other actives, that becomes more like a drug, right? So that becomes more like regular drug discovery once you get to that stage. But for us that are trying to deliver bacteria as a drug, there are some fundamental questions around how do you do that so these bugs actually stick around and do what they need to do immunologically. And I think anytime you see challenges within the field, you have to ask that fundamental question. Are those bugs sticking around? I use the term bugs because we do that colloquially. But this notion of using bacteria, are they sticking around and doing what they need to do? And so if you're using something that's wide transfer, how do you know you have the right ones? And then if you're using something very defined, how do you know you're getting them to the right place at the right time and enough volume?
So in animals, I believe, we've got animal models. First, you mentioned before in humans, you've got different states. So people have measured all kinds of different bacteria species in people with and without food allergies and identified differences. So then you say, okay, but is that just a correlation or is that causation? So then they do some experiments in animals. Tell us about those and what did they observe?
Yeah, and so they have done experiments in animals, but also people have done a lot of experiments in humans. So what we know is that FMT, which is what many people have, think about when they hear the microbiome and treatment, which is fecal transplant, which is the most, it's purified and it's not, straight poop transfer, but it can be. But that method is obviously not scalable and very difficult. It's donor derived and you're transferring at the end of the day. That is not what the biotech companies are trying to do at this point. So at a very minimum, they've taken a subset of that and said there's a wide consortium of bacteria that you need, and it comes from a donor, but these are now taken and a subset of that will be transferred.
I just want to make sure I understand. They've done fecal transplant studies, taking poop from somebody without food allergies and putting it into somebody with food allergies to see whether it changes their allergies.
So
and what did they find?
Those are unpublished at this time, but I can say that the results are promising, both for Pareto's key hypothesis, but also that's just the general notion that the microbiome is involved, and by rehabilitating the microbiome, if you will, that you can maybe have a therapeutic response. So
okay,
Rima Rashid out of Boston Children's Hospital has led some very small FMT studies in adults, and will be publishing those results.
Cool. And what allergens are we talking about? Is this more for wheat, gluten, celiac, or is this for, peanut?
It should be non-antigen specific. So the underlying, and just to get into the core hypothesis for Pareto's therapy, we believe that there is a key regulatory cell that is depleted in food allergic patients. So a very specific Treg type. So just for your benefit, I know you know what I'm, So this is a key immune cell type that is involved in helping to regulate your immune system. And our scientific founder, Talal Shatila, one of three that were involved in the discovery of this company or the development of this company, have identified a key regulatory cell that is depleted in food allergic children that is not that is present in healthy children. And through a series of both taking fecal samples from food allergic children and non-allergic and putting them into different animal studies, so this time mice, able to show that in different mice models, those that get the food allergic fecal transplant develop a food allergic type profile, and those that get the healthy develop healthy. So again, now taking this human understanding or observation, putting that into a mouse model, and say actually the microbiome or something in that transplant had a role in the development of this disease. Then through a series of experiments, looking at different mice studies, being able to show that in treating with very specific bacteria, you can actually reverse that effect or prevent it. And so in robust mice studies, and this has been published in Nature Medicine, they found that this specific Treg mechanism was involved also in these mice. So recapitulating the same key mechanism that was involved or observed in humans, now looking in mice and being able to see that intervening with a beneficial set of bacteria can actually prevent and treat that turning up these.
What does food allergy look like in mice? How do you know they're having an allergic reaction?
So this is there are different mice models and translational models. We could have a whole discussion about that. In this case, this is a F709 mouse that has been basically its IL-4 gene has been amped up. So it expresses high levels of IL-4, which kind of recapitulate key pathways involved in food allergy. And then what you do, what you're able to do is sensitize these mice through oral gavage using different antigen to become basically sensitized to that to that antigen and recapitulate anaphylaxis upon challenge. So what you're basically getting the mouse to do is become allergic to the thing that you're giving them. In this case, many times it's ova, but you can also do this with peanut and other food allergies.
What does an anaphylactic reaction look like in a mouse?
They lose, major temperature drop, diarrhea. They behave differently. They if you have extreme reactions, they can, they could succumb as well.
Okay, yeah. So just the development of a model like that is what enables this kind of research. Like that's its own breakthrough. And since it's nice to draw connections for listeners, IL-4, like ramping up the IL-4 levels in mice, makes sense if you consider that one of the drugs that we're all looking forward to seeing how it might work in dampening down the immune responses to allergens is a drug called Dupixent, and it's an anti-IL-4 antibody, IL-4 receptor, I believe, antibody. So we're looking to treat human food allergy by inhibiting IL-4, and so you're making mice food allergic by jacking up their IL-4 levels. And that starts to make sense.
And I realize you've kind of gone down a rabbit hole of kind of me talking to you as somebody who understands all of this. When we'll keep going, but I'm going to take a step back and I'm going to simplify kind of what we know and don't know also for the audience here, because I realize we've kind of gotten down into some weeds. But just to continue on the mechanism story. So this was the foundational discovery. And this is actually one of the foundational discoveries within food allergy as to our understanding of the role of the microbiome in the development of this disease. One of the other interesting links, if you just kind of take the compendium of science out there, relating the microbiome to any disease and the development of your immune system, and then what we're finding in food allergy and what we understand about the development of food allergy, is this notion of early life development. So there's another linkage here between what might the microbiome be doing within your gut environment that's leading to this proliferation of food allergy. Well, we know that food allergy develops very early in life. We also know that even food, immunotherapy, like peanut immunotherapy, works better earlier in life. We also know that prevention through food introduction is possible. So something about just perturbing the gut environment early in life can change your immune trajectory. Within the microbiome field, independent of food allergy, what we know are just fundamentally the microbiome develops very early in life. So although you have perturbations and changes to that over the course of your lifetime, what is also true is that it gets pretty set and it loses a lot of its plasticity in terms of its diversity and composition around 3-4 years of age. So even maybe earlier. So there's this notion of the first 1,000 days of life having a profound effect on the development of your microbiome. And also, independent of that, we know that your immune system has a plasticity that develops early in life where your immune system is learning. We all know this. And that linkage of the microbiome development with that immune development, combined with what we see happening in food allergy, is a little bit of a 1 + 1 + 1 that, gut health or microbiome health leads to good immune health early in life. And both of those are linked to something being disrupted in that is leading to this increase in food allergy. And so this restoration of the microbiome, could you actually really help turn the tide of what we're seeing in terms of the influx or the increase in prevalence of this disease state? And I thought that was something that was compelling around really trying to go for it in terms of the role of the microbiome, because we're not trying to just make a treatment and another drug therapy. We do fundamentally believe it could have potentially curative or preventative type effects, maybe as a monotherapy, but also in combination, but maybe a combination with food, right?
Now, you mentioned before that it's what you're really trying to do is to sort of get people's T regulatory cell profile to look more like what people without food allergies have. But what you're giving as a treatment are bacteria. So what's the connection between these bacteria and the Tregs?
So those pathways are harder to elucidate, but there's clearly, there's a lot of evidence that we could share with you, and I could go through in more detail. But at a high level, both seen in human patients, but also in all these translational mouse studies, is that the protection of or prevention of food allergy seems to be dependent on the prevalence of these Tregs, and that the Treg the amount of Tregs that you're producing seems to be mediated by the microbiome. And without getting into further,
the bacteria in your gut are influencing the kind of T cells that you have in your bloodstream.
There's evidence to suggest that there is some role in class switching of your of T cells and B cells. So you also have more IgE produced in these patients compared to IgA. When you are able to treat with the bacteria and either have, let's say, have a treatment effect, you're able to see that ratio change. So that class switching, is that a byproduct of increased Treg production, or in and of itself, is there also a mechanism involved with class switching of IgA and IgE? There's some really interesting work going on even within that field at Yale. So Noah Palm within our world, Stephanie Eisenbarth is doing some interesting work in IgA. And the role of the microbiome, is also very, I think now well observed that it has some role in class switching of IgA. So there's another, there are many mechanisms that we think that are involved. It's not a one-to-one, but the primary, marker, if you will, that we see is this induction or increase in this key T regulatory cell. Yeah. And also, if you think about what is known around immune tolerance mechanisms more broadly, just outside of food allergy, if you think about the framework, we know that you have to have some exhaustion of kind of the pathogenic T cells and B cells. You need to be able to switch those to kind of more, therapeutically beneficial or non-disease, or pathogenic in nature. You need to have some kind of depletion of the memory of those cells so you don't produce them again in the future. And then you need to maintain that through some type of regulatory mechanism. So by turning on that regulatory mechanism, that's why, as you know, there's so much interest in regulatory mechanisms that could be more durable in their effect if you can actually get them to be, produced in a durable way. And when you think about the microbiome, maybe that's one way that you could do that.
So I got a couple questions, and we just have like 5 minutes or so. So let's do them kind of quickly. There's one question that I like to ask people that are working at the cutting edge, where you have so much evidence for why it should work. Let's say we're several years down the road. First of all, when will we know whether this works? Is this like a year away, 2 years away? Like how far?
We would like to get into our phase 1 study next year and go directly into food allergic children, and hopefully have enough powered in our phase 1 A/B design that we could get some signal of positive trends of activity.
So plausibly speaking, within like 3 years, maybe earlier, you ought to have a result that tells you whether you're on the right track or not. So if we come back together again in 3 years, I'm sure we'll talk before then, and nothing worked, and you look back and you think, how could it be with all of the all the evidence that I had going into this, how could it not have worked in humans in these studies? What might be the explanation that you look back on and say, maybe this was the assumption that was wrong?
A lot of ways I can answer that, just to keep it simple. How do you know that you had enough of the bacteria administered to the right part of the gut for the right amount of time? So that dosing question, essentially, for what is a drug, in this case a set of bacteria, is incredibly complicated. And that's where a lot of the fun science is also evolving in terms of the microbiome field. But the question of, did you deliver it at the right time? And let's just take even infants, because we are hopefully getting down into an infant population if we're thinking about prevention. When do you do that? And how do you do that in conjunction with what is already there in the gut environment and what is coming externally in terms of other food consumption or other non-endogenous factors that might be affecting that environment? So that becomes a whole complex issue. And then that's separate from a whole other fundamental set of questions around subpopulations and potentially subgroups that may be more relevant and not, because certainly not all of the increase in food allergy is purely based on just this gut microbiome phenomenon. That may be partially driving the rise, but certainly there have been people with food allergy prior to some of the environmental impacts that we've done to ourselves. So, I think that thinking about subpopulations, which I know you've talked to Wayne and others, that's a whole other field that you have to overlay on top of this as well.
Yeah. So it's cool to call your pocket basically and say, like, if we fail, it could be for these reasons. Because sometimes people are really quick to say, oh, negative result. Yeah, okay. So that line of inquiry is dead, that field's dead, and they don't come back to it. And yet, people have to have the courage to go back and say, no, let's try it differently this time. Right. And certainly the field of Alzheimer's is riddled with failure. And, people stay with it. And occasionally they achieve a breakthrough in doing something different, slightly different than how others did it before. And then people realize, oh, I see, there always was, some sort of crack we could exploit. Maybe now we've found it. Last question. If there were price controls on FDA approved, medicines in the future, anything that, required insurance reimbursement, would you still be able to raise the funding that you need to do this? Would you be able to pursue your R&D or not?
You tell me.
Well,
I'm joking. So I'll give you my answer. I think yes, and I'll say that because this is partially personal in terms of just the kind of things that I want to be involved in, but also specific to the opportunity that we're trying to develop with Pareto. It's not an incremental impact, so let's try to create real value for patients and real value, ultimately to the healthcare system. And let's figure out the economics. But certainly with the size of the food allergic population, the size of the population in this case, unless we get to some really subtype that we're going to target in the future, we're talking about a lot of people that are afflicted, and we need to think about prices that are commensurate with that population size and the impact that would have on the healthcare system.
Sure. 8% of the population has food allergies. So, you're dealing with what, 10, 20, 30 million people in the US alone. conceptually, if you could get this to everybody, you could charge certainly under $1,000 a year, and you're looking at what, $30 billion. Like, but of course, nothing ever gets to 100% of people. And so, who will you get it to? If this works only in infants and you got to give it in the first year of life, now you're talking 4 million births a year. And are you really going to, talk mothers into giving this to their babies? So maybe if you get it to even 10%, that would be quite remarkable. So now 400,000 babies treated. Well, now what do you have to charge? And will insurance reimburse for that? So I don't know, like,
so that's a very good, this is a really interesting question from an industry and a societal impact perspective. I know we're going to run out of time. I think that the other side of this, I did watch your last podcast, and I think that the aspect of healthcare, organizations having innovation groups is fascinating. I agree with you. It's very sad to hear their horizons in terms of the timelines. And I think that challenge fundamentally that you've raised is the one that we should be thinking about, not just in terms of how do you allow for innovation to invest, to create amazing breakthroughs that should be, valued, but then do that in a way that it's not going to destroy, society in terms of the value. And I think it's a very hard question.
Well, food allergies are already destroying society, right? It's
Sure.
already costing us tens of billions of dollars a year, right? Plus it weighs on our minds. And I don't even think people have even calculated what the cost of the anxiety is of families that are worried, that their next child will have food allergies because they already have that in the family, or when their child is diagnosed with it, what day will be the day when they discover that their child accidentally ingested some, you sit on a plane and they're not careful about making sure that there aren't tree nuts and peanuts and whatever on the airplane seats, right? And you're up in the air and they have an anaphylactic reaction. Like, these are the things that you carry in your mind. So I would argue we're already paying a price, a huge price that often isn't quantified. I'd be curious, when in the course of developing your product, will you do the math to calculate what the societal value is of the innovation that you hope to bring, to the market, in order to anchor, payers and the public and the media and anybody who might lambast the price of your product someday, anchor them to, oh wait, it's worth a lot. And then later down the road, when you do set a price, they might be pleasantly surprised to discover that actually the market, if there's competition or whatever, oftentimes ends up paying a price much lower than the societal value of that innovation. So is cost effectiveness something that you think about doing along the way, now because of the talk of price controls? Because it's certainly not normal for development stage companies to do that kind of math.
I think about access and cost, effectiveness all the time. And have been so immersed into, the industry's trend of thinking about these considerations earlier in the life cycle of products. So I think there's both the theoretical model that you could develop now. A lot of those assumptions, as you know, need to be better refined with data as we proliferate kind of some of the initiatives that FAIR is actually involved in terms of data collection. And finally, as you think about kind of, a single indication opportunity, if you will, for a drug, and take us out of it, just any drug developer, if there are incremental populations that you can go after, we all know that that's possible, but also creates more R&D cost. How do we actually reduce that and make it more efficient then with data and real-time reporting and FDA policies and the healthcare system? I think that is, something that has been talked a lot about in the past. There's all sorts of barriers as to why that can't happen, seamlessly, but we're going to have to solve that if we're going to really try to keep innovation going. And that's something that I think that, I'd be happy to have discussions with, any stakeholder related to how to do that better now.
All right, Tag, you're it. As the former and, forever, FAIR chief strategy, thinker. Kari and I spoke during our episode, I believe, about the need to lead the cost effectiveness analysis of increments of progress on food allergies, and make sure that the math is fully developed to capture all the ways in which this kind of innovation would help, so that in the future, we're not going to be told by groups like NICE in the UK or ICER here in the US, based on oversimplified, really like anemic math, that, well, this doesn't seem that important, right? Well, sure, you ignored a whole bunch of features. You ignored the anxiety that, you know, everybody lives with. You ignored that drugs go generic, et cetera. How do we take charge and make sure that the value proposition is one that we, the, you know, the stakeholders, the, I don't know, food allergy warriors. Maybe I shouldn't have called you an expert. I should have called you a food allergy warrior, right? Like that. How do we make sure that we put out this framework so that others who don't care as much don't put out in a framework that's destined to underestimate the value of progress? So let's put our heads together. And at some point, I would love for us to actually run one of these projects. You know, it'll cost several $100,000, but we know who to hire to do this. I think Kari said that there are people at Stanford that could do it. So maybe we can pull on that thread. We'll see where we get to. But come together and start to be able to plug in, if, you know, Pareto's product offers a certain magnitude of benefit, how much of, you know, the problem has it solved, and what's that worth?
Yeah, absolutely. I think about those things a lot. I mean, not just for product development, but for the reasons that you're describing. And it leads to some very interesting, I mean, we won't go through it now, but it obviously will, to get to that framework, that's obviously possible. You know, you've seen those analyses before, and we can start building towards that and make it better as we, and kind of make that a living framework, if you will, that we can improve on. I absolutely agree that, you know, having that would be like critical, just for all the reasons, communication and foundational knowledge as well. It then opens the question of then funding that innovation and not having the lost innovation, and how to do that becomes the second question, you know, in terms of the economics, right? Because
I'm just going to throw out an idea, you know, proper insurance, collectively, you know, make all this affordable. But Nurry, thank you very much for joining for this episode. And I'm sure we'll continue to pull on a whole lot of threads together in the food allergy space. Thank you.
really appreciate it, Peter. Thank you for your time.